Bible and my Swedish Bible, so it's been some confused. Yes, in the Psalms. Yes, I realized that today too.
In German.
Yeah. Okay. Okay. Ja, välkomna allesammans till den här lilla morgonandakten. Nej, just det, du räknar ner också. Nej. Ja, men ni är alla varmt välkomna, ni som har kommit hit till den här morgonandakten. Jag tänker att vi ska sjunga ett par sånger, jag ska läsa ett bibelord också. Ska vi be för den här dagen, och som är vår sista dag på den här Genesis årskonferens. Vi ska sjunga Vila i nåden, och det är nummer 10 i lovsångshäftet som ligger där nere. Vila i nåden, bli helad i det. Låt honom krama om dig, tycka om dig som du är. Vila i nåden. Vila i nåden, bli helad i den. Låt honom krama om dig, tycka om dig som du är. Varje morgon väcker han dig med en stilla röst. Mitt barn, jag vill umgås med dig. Vila i nåden, bli helad i den. Låt honom krama om dig som du är. Varje morgon väcker han dig med en stilla röst. Mitt barn, jag vill umgås med dig. Vila i nåden, bli helad i den. Låt honom krama om dig, tycka om dig som du är. Hans barn, hans barn, hans barn. Jag ska läsa ifrån psalm 30. Psalm 30. Och det är David som har skrivit den här psalmen. Jag vill upphöja dig, Herre, för du drog mig upp ur djupet och lät inte mina fiender triumfera över mig. Herre min Gud, jag ropade till dig och du helade mig. Herre, du förde min själ upp ur dödsriket. Du tog mig levande bort från dem som går ner i graven. Lovsjung Herren, ni hans trogna. Prisa hans heliga namn. Hans vrede varar ett ögonblick, hans nåd hela livet. På kvällen gästar gråt, på morgonen kommer jubel. Jag sa i min trygghet, jag ska aldrig vackla. Herre, i din nåd gav du styrka åt mitt berg, men du dolde ditt ansikte och jag blev förskräckt. Till dig, Herre, ropade jag, till Herren bad jag om nåd. Vad vinner du på mitt blod, på att jag går ner i graven? Kan stoftet tacka dig? Kan det förkunna din trofasthet? Hör, Herre, och förbarma dig över mig. Herre, var min hjälpare. Du förvandlade min klagan till dans. Du lossade min sorgdräkt och klädde mig i glädje. Därför ska min ära sjunga till dig utan att tystna. Herre min Gud, jag vill tacka dig för evigt. Jag sa det igår lite grann att jag ofta man kan läsa Bibeln med lite olika glasögon och då talar texten till en på olika sätt. Och jag tänker att om du tänker att Jesus läser den här psalmen, det handlar om hans liv på något sätt. Vad han har gjort för oss, han som har varit i dödsriket för våran skull. Och det står i vers 6, hans vrede varar ett ögonblick, hans nåd hela livet. Och jag tänker att det där ögonblicket av vrede, det var ju när Jesus hänger på korset och låter all vrede, all synd, all skam drabba honom. Gud själv tar på sig det. Han tar på sig all vrede för våran skull. Ett ögonblick, en dag i historien som är helt avgörande. Den här långfredagen då Jesus gav sitt liv för oss. Hans vrede var ett ögonblick. Och sen står det att hans nåd hela livet. På grund av det som Jesus gjort så finns det nåd för hela livet. För hela universums framtid på något sätt. På kvällen gästar gråt, på morgonen kommer jubel. Vi vet kvinnorna som kom till graven på söndag morgonen. De var ledsna. De tyckte allt var kört. Men de mötte en tom grav. Han var levande och han var uppstånden. De mötte Jesus. Det var seger. Du förvandlade min klagan till dans. Du lossade min sorgedräkt och klädde mig i glädje. Därför ska min ära sjunga till dig utan att tystna. Herre min Gud, jag vill tacka dig för evigt. Så låt oss göra den här morgonen. Tacka Herren för vad han har gjort för oss. Låt oss tillsammans be för den här dagen och för föreläsningen som vi snart ska få ta del av här. Och för gudstjänsten vi snart ska fira. Vi ber för Nadja som ska hålla föredrag och för Göran Schmidt som ska hålla predikan sen i gudstjänsten. Och det kommer ganska många människor också som kanske inte har tagit emot Jesus personligen. Så låt oss be att människor får bli frälsta den här dagen. Det vore ju fantastiskt därför att vreden har drabbat Jesus och nu finns det nåd för varenda människa. Herre, jag tackar dig för den här dagen. Jag tackar dig för att vi får börja med att läsa ditt ord och lovsjunga dig och tacka dig för vad du har gjort för oss. Tack för att du gav ditt liv på korset. Tack för att all vrede drabbade dig, det som skulle drabbat mig och oss alla. Jag tackar dig för det, Herre. Tack för att du hade en plan, Herre, en frälsningsplan redan från början när du skapade universum, Herre. Och tack för att vi idag får leva i din nåd på grund av att du har frälst oss, på grund av vad du har gjort för oss, Herre. Och jag ber för den här dagen, för de människor som kommer hit till Immanuelskyrkan här i Malmö, Herre. Jag ber, Herre, att de skulle få möta dig som den levande uppståndne, Herre. Du ser de som kanske ännu inte har tagit emot dig, att de skulle få bli frälsta idag, Herre. Få uppleva vem du verkligen är, Jesus. Jag ber för föreläsningen vi snart ska få ta del av. Jag ber för gudstjänsten och predikan, Herre. För sång och musik, allt det som ska ske idag, Herre. Och jag tackar dig för de här dagarna vi har haft tillsammans här på konferensen och ber att vi ska få en riktigt fin dag och få uppleva din närhet idag, Herre. Jag ber om det i Jesu Kristi namn. I Jesu Kristi namn. Amen. Tack Jesus. Tack Jesus. Vi ska sjunga en sång tillsammans, tänkte jag. Du är med mig, nummer 6 ska det vara i häftet. Vi har sjungit den här mycket i våran kyrka, nästan blivit lite som en temasång för oss på något sätt. Du är med mig, nummer 6 i häftet. Du är sanning som står kvar, Jesus. Du är hopp för nya dagar, Jesus. Jag kan lita på ditt ord, Jesus. Du är grunden där jag står, Jesus. Du är med mig, bredvid mig i elden. Du är med mig i dödsskuggan dödsskuggans dal, alltid med mig. Ge frid mitt i stormen, Jesus, Jesus. När jag vacklar står du kvar, Jesus. Styrkan när jag inget har, Jesus. I din kärlek vilar jag, Jesus. För din nåd är ny var dag, Jesus. Du är med mig, bredvid mig i elden. Du är med mig, i dödsskuggans dal, alltid med mig. Ge frid mitt i stormen, Jesus, Jesus. Du är med mig, bredvid mig i elden. Du är med mig, i dödsskuggans dal, alltid med mig. Ge frid mitt i stormen, Jesus, Jesus. Kristus med mig, Kristus framför mig, Kristus bakom, alltid nära. Kristus i mig, Kristus under mig, Kristus över, alltid nära. Kristus med mig, Kristus framför mig, Kristus bakom, alltid nära. Kristus i mig, Kristus under mig, Kristus över, alltid nära. Du är med mig, bredvid mig i elden. Du är med mig, i dödsskuggans dal, alltid med mig. Ge frid mitt i stormen, Jesus, Jesus. Du är med mig, bredvid mig i elden. Du är med mig, i dödsskuggans dal, alltid med mig. Ge frid mitt i stormen, Jesus, 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 Jesus. Tack så mycket.
There we go. And then we'll have to try to quickly switch. So you're on?
Kan jag börja nu eller ska jag vänta? Ja. Ja, välkomna allihopa. Jag heter Ola Hörscher och jag representerar Genesis, Skapelsekonferensen. Och vi är väldigt tacksamma från Genesis att vi har fått vara i Immanuelskyrkan här i Malmö. Vi hade en forskningskonferens torsdag till fredag om skapelsetro. Och sen från fredag kväll fram till nu på söndag har vi haft Genesis årskonferens här. Och vi har flera inbjudna föredragshållare från olika länder. Och det har varit otroligt spännande att lyssna till. Och nu ska vi ha ett sista föredrag nu på förmiddagen och sen så kommer vi att ha en gudstjänst tillsammans. Och Göran Schmidt från Genesis kommer att hålla en predikan under den gudstjänsten. Och jag är väldigt glad nu att introducera Nigel Crompton som kommer att hålla ett föredrag. Nigel Crompton, han är professor i biologi vid Cornerstone University och han tog en kandidat- och masterexamina. Han växte upp i Storbritannien, så han tog de examina vid Manchester University. Han doktorerade i Tyskland vid Justus Liebig University i Gießen. Och han tog en andra doktorsexamen i Schweiz vid Zürich universitet. Och i USA då, vid Cornerstone University i Michigan, så undervisar Crompton i biologi, genetik, molekylär cellbiologi, bioetik och ursprungsfrågor och neurovetenskap. Och Nigel Crompton har publicerat mer än 100 vetenskapliga arbeten. Och han är också medarbetare i den tyska skapelseföreningen Wort und Wissen. Och idag kommer han att hålla ett väldigt intressant föredrag om att man kan ha väldigt snabb artbildning, livets buskmodell. Att Gud skapade arter efter sina slag. Alla hundar har gemensamt ursprung. Och redan Gregor Mendel på 1800-talet förstod att man kunde ha snabb artbildning inom varje buske. Och det ska Nigel Crompton prata om. And so, Nigel, we're very glad to have you here and looking forward to hearing your speech or your talk on Mendelian speciation and the origin of species. So, welcome.
Thank you.
Thank you so much everyone for coming today. Really appreciate that. I hope you get a lot from this lecture. It would be a shame if you didn't. Perhaps just to start, it would be nice to say a word of prayer. Lord Jesus, thank you. You're with us. You love us. Lord, open our eyes. Help us to see you. Help us to see something of the wonders of your creation in this lecture today. Thank you, Lord Jesus. Amen.
Amen. Just quickly, is there någon som behöver översättning från svenska, så skulle jag kunna göra det i något hörn eller något?
Jag kan ju översätta om du vill. Jag måste tänka på.
Ja, precis, det får vi göra.
Är det någon som vill ha översättning? Ja. Räck upp handen i så fall om det är någon.
If I understood more Swedish, I would know what was going on.
He's asking for translation to Swedish if someone needs it. So,
Same problem, but the other way around. So thank you very much. I would like to talk about Mendelian speciation and the origin of species. And I want to start this talk by looking at a very famous book by Darwin. It's called On the Origin of Species by Means of Natural Selection. The title actually goes on, it's quite a long title. Or The Preservation of Favoured Races in the Struggle for Life. However, if we look at the first part of the title, On the Origin of Species by Means of Speciation, what Darwin is saying is that species arise via natural selection. And this idea has become ingrained in our society, but it is incorrect. Many folks get confused about what natural selection is due to the notable esteem granted to Darwin's idea. Yeah. They seem to imagine it's an enchanted process, this natural selection, yeah, which can cause new species to magically appear. If you watch fabulous television programs by describing things by naturalists, and they'll be talking about some wonderful animals, and then they'll suddenly say, oh, well, natural selection gave rise to this, or natural selection gave rise to that. But natural selection, all it is a form of selection. Yeah? It selects things which are favoured, yeah? But it's not magical. It can't call anything into existence. Yeah? If these species aren't already there, it can't select them. It's just not possible. Yeah? There are other forms of selection. There's sexual selection, there's artificial selection, and the same thing applies. If these species, if these phenotypes aren't present, they can't be selected. Natural selection can't bring something out of nothing. And therefore we're left with alternatives. What are the origin of species? Mutation. And what I'll be describing to you today was actually the answer which Mendel gave even when Darwin was just becoming famous through his book. And so I'd like to share those ideas with you today. The goal of the talk is to examine the origin of new species, but based on field observations. What do we actually see? Not what is speculated in textbooks. In fact, hypothetical mechanisms of speciation based on speculative roles of mutations, they overwhelm our biology, genetics and evolution textbooks. But this is all speculation. To be honest, trying to account for novel species, for new species, by mutation or by altering existing species' various genes, it's rather like trying to produce a new book by mutating or altering an existing novel. Just changing the words and making a new book out of it. It's not a very promising approach, really. Mendel's studies were conducted standing on the shoulder of giants. He readily acknowledged his debt to various other well-known botanists. His paper is fabulous to read. It's a lot of fun. It's available on the web, on the internet. And if you can speak German, it was written in German, the fabulous English translations. It's really well worth reading. It's just a lot of fun. Yeah. But he acknowledged these well-known botanists, people like Gärtner and Kohlreuter, and they'd spent their lives looking at hybrids. And one of the observations that they made was that hybrids tend to revert in their appearance to the parental form. Yeah. It was a well-known phenomena, and Mendel called it das Entwicklungsgesetz, the law of development. Now what he meant by that was the law of population development, not embryological development, but the law of how populations develop. That's what he actually meant by that. And this law resulted in systematic genetic changes, and they occur in hybrids. I'll tell you a little bit more about them later. And Mendel understood it was caused by something called loss of heterozygosity. Now, for many of you not familiar with genetics, I will be explaining a little bit more about that later. When we talk about loss of heterozygosity, actually what happens is instead of heterozygosity, we end up with homozygosity. So loss of heterozygosity, that's the same thing as gain of homozygosity. If you're not familiar with those terms, I will be describing them a little bit later. So don't give up yet. A statement repeatedly heard is that speciation is caused by mutations, DNA sequence changes. Now, in one sense, it could be true. Yeah? If I want to change a car repair manual into one of Shakespeare's plays, or into a biology textbook, or into a theological exposition, yeah, I could do that. And the way I could do it is I could change the letters one by one, yeah? And they'd probably have to change the number of letters, but eventually I would sort of have some sort of limited success, and perhaps by doing it long enough I could finally get then the complete works of Shakespeare, whatever. But this is not how speciation works in nature. And we need to be very careful when we talk about mutations causing new species. We have to understand the implications of that. Here's a whole bunch of humans, some of whom have rather unusual mutations. On the top left, there's someone there suffering from hypertrichosis. It's also called the werewolf syndrome. He's a lot of additional hair, prominent hair, all over his face. But he's still very much human. Or the young lady below him, if you check out her eyes, you'll see she has heterochromia. One eye is brown, the other is blue. It's caused by a mutation. She's still very, very much human. Yeah. The young gentleman below her, he has piebaldism, so a patch of white at the front of his. It's also caused by a mutation. Technically, redheads, yeah, are caused by a mutation. Yeah. The young gentleman at the bottom right, he has a columba, cat's eye. These are all clearly human. There's been no speciation has taken place here. You can take the fruit fly, Drosophila, and you can mutate it. And you can mutate it and get all sorts of changes in appearance. But it remains the fruit fly, Drosophila melanogaster. Mutate as much as you will, you always get the same species. Similarly, alter the rate of gene expression as much as you will, and you always get the same species. Mutations degrade genetic information, epigenetic modifications or transposition. They can be programmed to change traits, the things we see, but it doesn't give you a new species. Now, don't get me wrong, epigenetic modifications or transpositions can result in the expression of hidden information, so they're very important. They make significant contribution to change. Yeah. But they're simply players in a larger scheme. So what's going on here? If not mutations, then what? One can ask two very practical questions. Can we repeatedly make a new species? That's an interesting question. Can we actually do that? And if we do, then what was the genetic mechanism involved? So let's have a look at some examples. There are very, very many examples amongst plants. These are just a few agricultural examples. The grapefruit is a hybrid, cherries are a hybrid, bananas are hybrids, peanuts are hybrids. They're their own species, but they were caused by hybridization. Let's look at some fun animal examples. So here's the Old World tufted duck and the Old World pockard. Old World because, of course, they're Eurasian, down in Africa. If you cross them, you get the Ithira hybrid here, which, if you see at the bottom, is exactly the same as a bird you find in the Americas. A new species via hybridization. There's another example. The shoveler and the cinnamon teal, if you cross them, the hybrid's at the bottom. Here is the Australian shoveler that's quite in a different area, and you see it's the same species have been caused by hybridization. And what is fascinating is this white eye patch here. That white eye patch is something new you didn't see in the parents. Information was hidden but became revealed. That's a very, very important aspect of Mendelian speciation. Here's some lovely little mannequins, sweet little green birds that hop around in the Amazon, in the trees, dicky birds. And you can see the three ones here. The one in the middle, the golden-headed mannequin, is the hybrid, but it's a separate species. In North America, the blue-winged warbler and the golden-winged warbler, if they form the hybrid, which is then the Brewster's warbler, and that can hybridize with the chestnut-sided warbler to produce Burkitt's warbler. And we're seeing all these species. Very recently in a prominent science journal called Nature, there was an article about the grey snub-nosed monkey. And they just discovered it was actually a hybrid between the golden snub-nosed monkey and technically an ancestor of the black snub-nosed monkey. Now, for many biologists, hybrid species are something of a paradox, because prominent leaders in the field of biology, folks like Dobzhansky and Meyer, they have persuasively argued for decades that species are reproductively isolated. And if species are reproductively isolated, they can't form hybrids. And if they did form hybrids, well, then actually it was that the hybrids were sterile. That was the argument. And so biologists had a tough time accepting that. But as soon as it became obvious this wasn't really correct, hybrids were found all over the place. It only turns out to be true that species are reproductively isolated about half the time. Now, this reproductive isolation is a very important mechanism in speciation. So it's a paradox. There's some truth to it, but you still get hybrids, and many of these hybrids are new species. How can I change many genes all at once? If I could change many genes all at once, I would have lots of new traits. That would start to look a little bit like a new species. But how could that happen? Well, it actually happens during meiosis, during the process when germ cells are made and produced. At an early stage of meiosis, during Pachytene of prophase 1 of meiosis, during early stage of meiosis, all of the chromosomes line up, the homologous chromosomes. You remember we got chromosomes from mum and chromosomes from dad? Well, those chromosomes they line up, and they line up exactly. There's a very special machine that does that, a synaptonemal complex, and of the 100 million base pairs in these chromosomes, they're lined up exactly. They have to be. If it was lined up inappropriately, it gives all sorts of problems. It's very fascinating that can happen. And then you can exchange genetic material. But you're exchanging lots of genetic material, and so you're exchanging lots of traits. And so now we can see how we can get a new species. Let's have a look at some hybrids, shall we? So the hybrid between the horse in the top right and the zebra at the bottom left, you get either a Hebra or a Zorse. It all depends who was the male, which one was the male parent, which one was the female parent. Yeah, and the names here, they're sort of rather exotic, Hebra and Zorse. It's an indication of that the first part of the name reflects the male parent, and the second part of the name reflects the female parent. So a Hebra is a male horse and a female zebra, but a Zorse is a male zebra and a female horse. And that's why they get these funny names. Yeah. And you can see that fascinating animals, these hybrids. What tends to happen is the hybrid looks a little bit more like the male parent than the female parent, interestingly enough. Another famous example is the cross between lions and tigers. Yeah. And so we get the tigon, where the male parent is the tiger, and the liger, where the male parent is the lion. Ligers are very, very fascinating animals. They lose growth control and they become absolutely enormous animals. Huge animals, very, very fascinating. Well, what about a swan and a goose? You can get the swoose, which has the male parent there is the swan, or you can get a guan, and the male parent was the goose. Here's a slightly more exotic example. These are birds of paradise. And we have here the kingbird of paradise and the magnificent bird of paradise, and they can give rise to these two different hybrids. And if you look at the tail, you can see that one's a little rounder than the other, and you can work out then who the male parent was. Well, what do these hybrids tell us? First of all, they tell us that a species is a set of, we refer to these things as characters. Yeah, it's what we observe. Yeah. Two copies of each character are present because we got one from mum and one from dad. Yeah. The characters can have alternative traits. They could be the same, but they can also be different. Yeah. And usually, only one trait at a time is expressed. So the one that is expressed is called the dominant one. The other one is, so the information is still there, it's just hidden. It's a very important point. We'll come back to this a little bit later. And novel combinations of what's expressed, that produces novel phenotypes, that produces novel species. The information for speciation is already present in the genome. Now that's an interesting point. We don't have to mutate anything into existence. Everything we need is already present for all of these different species. I'll come back to that in a moment. First of all, I want to mention some very spectacular examples of speciation. These spectacular examples, they're referred to as adaptive radiations. And they include things like plant alliances, lots of different plants on Hawaii. Another example are the picture-winged drosophila, the fruit flies in Hawaii. Then there's fish, the cichlid fish, in the Great Lakes of Africa, the East African Rift Valley lakes. We've got then lizards, fun little lizards, in the Antilles, in the Caribbean, in the big islands of the Caribbean like Cuba and Jamaica and so on. And also finches. Perhaps the most famous adaptive radiation are Darwin's finches on the Galapagos Islands. But if you go once again to Hawaii, and I would encourage you to do that, particularly in the winter here in Scandinavia, you might want to take a holiday in Hawaii. The finches on Hawaii, they're much more spectacular, far more fascinating. Well, let's start with this little guy, shall we? A little bit like a sort of a daisy with a yellow florets around the outside rather than white. It's called a tarweed. It's called a tarweed because it has sticky secretions on it. And you find these in California, quite small. This particular example is very important for if you want to find out if that person you happen to be rather sweet on likes you or not. You should definitely use this flower because it has one petal. So she loves me. Yeah. And here's a second species. This is also another California tarweed. This one has no petals on, no ray florets on, none of those big petals around the outside, just on the inside. And the Pacific golden plover, it's a bird, took these seeds, they perhaps got stuck to the feathers or whatever, and flew them across from California over the Pacific to Hawaii. And they crossed there. And then this is the allopolyploid. When they cross, this is the chromosome cross, you can actually see it. They produce then a plethora of plants. Now these tarweeds, they're relatively small plants, you saw them, but they produce then all of these different species. They produce these silver swords here, fabulous plants. They're called silver swords because of their leaves, which are covered in little hairs which reflect the light. And so they look silver. Because they're long and narrow, they look like swords. And that's usually all you see. And they look sort of like massive hedgehogs on the lava fields. Not the hot lava fields, obviously, when it's cooled, but those lava fields are sufficient for them to grow. And then once in their lifetime, they're monocarpic, once in their lifetime, they flower. And up pops this panicle with these sunflower-like flowers. They're not yellow, they're purple. To give you an idea of how large this is, if someone was standing next to it, they would be about as tall as this plant is. Well, that's one of the species that these two little tarweeds gave rise to. But they also gave rise to these extraordinary plants here, which are even more extraordinary when they flower, because you get something similar to a panicle like this popping out the top here. Magnificent plants. Or cushion plants or bushes. Lovely plants, fascinating radiation, lovely place to visit to check them out too. Or even trees from those little tarweeds. Whoa! All the information was there. It was just waiting to be expressed. These are the different species of the tarweeds shown in a circle, and the lines indicate where hybrids can take place. Now, hybridization means that the species are so closely related they can actually produce children. And you can see almost all of them can. This one can't, but then again, it's gone extinct. And it's very difficult to make hybrids from something which is extinct. So that's one example. They're the silver swords. What about the fish in Africa? This is one of the Great Lakes in Africa, Lake Tanganyika, and here's another group of fish from another lake nearby, Lake Malawi. They are separate lakes, and the fish arose independently of each other in the separate lakes. And even though they look like each other, all of these species are genetically more closely related to each other than any one of them is to any of these. These were separate radiations, and yet similar fish appeared. And that's telling us that the information is present. It was present from the get-go. It is present now, and it simply needs to be expressed. Another example here are these lizards from the Caribbean. And on the main islands of the Caribbean, you find these lizards. They arrived on each one of the islands independently of each other. They went through a radiation independently of each other, and on each of the islands they produced these different types of lizards. There's always the same kinds of lizards which are produced. It's very fascinating. These are called trunk-crowned species. This trunk species, it's very difficult to actually see it. Can you see the lizard? Yeah. They're called dewlap lizards. They have a fancy throat patch. And if they inflate it, it's brightly coloured red or orange. Yeah, then all the lady lizards go weak at the knees, yes, at the sight of this throat patch. Yes. But what is fascinating is that this happened independently on each of these 4 islands. The information was already in the genome. Nothing had to be mutated into existence. We have Darwin's finches. Very similar process took place. Presumably, first species arrived, and then it produced a lot of new species. Now these are, as I say, Darwin's finches. There's some difference between them, but if you go to Hawaii and check the finches out there, extraordinary differences. These are the honey creepers, the finches in Hawaii. The classic, the iiwi, is an icon of the island, lovely little bird. And there's all of these others, yellows and reds and all sorts of different colours, and the beaks, and huge differences in the size of the beaks. Now, 2 critical observations can be made based on all of this. First of all is that these radiations, these adaptive, they're constantly associated with minimal genetic change. If you look at the DNA and you sequence the DNA, and that has been done, I'll tell you a little bit more about that later, just a little bit more, but you find there's very little genetic change. So how do we get all of these new species if almost no genetic change is occurring? This is where Mendel helped, come back to that in a moment. And of course, these adaptive radiations, they demonstrate that the information was already present. There are genetic subroutines present in the genome ready to be revealed. How do adaptive radiations take place? How can we get major phenotypic change, new species, yeah, but with only minor genetic change? And Gregor Mendel, the father of genetics, he provided us with a simple, clear answer to this. He demonstrated that because genes have at least two alleles, we got one from mom and one from dad, because genes have at least two alleles, an enormous amount of phenotypic variation is available for profuse speciation. So here he is Mendel. He wasn't a monk, he was a friar. Well, that's got nothing to do with McDonald's. But friars, they still make this promise to remain celibate during their lives, but they get involved in society. They don't hide in the monastery. So he was a very prominent person in the politics of his particular town there, Brno, which is now in the Czech Republic. And while he was doing these studies with his peas, Darwin's book appeared. And a German translation was quickly made. And he got hold of a German translation, and that is known. It was found in his library in the abbey where he works. So what experiments did he do? So now let me try to explain a little bit about how it's possible for speciation to occur via this mechanism. So he took peas, garden peas, and he had peas which had purple flowers, and he had other peas which had white flowers. And when he crossed them, all of the progeny, all the children, had just purple flowers. Now this was very sad because the white flowers had gone. Nobody threw a funeral, but there were no more white flowers. That was very sad. When he took those purple plants, those children, and when he self-crossed them, suddenly in the grandchildren, white flowers reappeared. They hadn't actually been lost, they were just hidden in the genome. Now he did this not just for flower colour, but for 7 different characters. For flower colour, seed texture, pod colour, pod shape, flower position and plant height. And every time he had 2 different alternatives. The different alternatives are called traits, and it's what you actually see. Yeah. And always on this particular picture, the way it's shown is the one on the left is always the dominant one. So those were the traits which were seen in the children. And in the grandchildren, with a ratio of approximately 3 to 1, we started to see the recessive trait again. So how does that occur? Well, I don't want to go into too much. This is not meant to be a genetics lesson. I don't want to go into too many details, but basically, when he crossed his white parent with his purple parent, he got a hybrid. They were all purple. And when he self-crossed those, so here's the self-cross here, he got this 3 to 1 ratio. And what I want you to notice is that there was one character or one gene involved, but it gave rise to 2 phenotypes. So one gene, 2 phenotypes. So what happens if you do 2 genes? Well, he did that experiment. He did even 7 genes, but I won't go into all of that. Whoa! But with 2 genes, everything gets twice as complicated. And in this particular case, he has 2 genes, and if you look at these particular drawings, they're called Punnett squares, whatever, and you look at this diagonal here, this is the leading diagonal. What's very nice, if you draw them correctly, the leading diagonal reveals all of the different phenotypes. And so you can see very nicely here, 1, 2, 3, 4. So one gene gave 2 phenotypes, 2 genes gives you 4 phenotypes. Well, what happens if you do 3? So this is the 3 gene situation. I just labeled them A, B and C to make it too complicated. Once again, the big fancy Punnett square. If you look at the leading diagonal, there you'll see each of the different phenotypes you can get. And this time it's 1, 2, 3, 4, 5, 6, 7, 8. So 3 genes give you 8 phenotypes. I don't know how many of you are mathematically gifted here, but you might notice what's happening. As you increase the number linearly, the number of genes, the actual number of different phenotypes increases exponentially. So very quickly you start to get very many differences. So if you have just 9 genes or 9 characters, you can get 512. And if you have 20 genes or 20 characters, that's all, that will give you 1 million different phenotypes. Well, there is no family on Earth which has 1 million different species in. But the potential's there. The potential's there. That's very fascinating. And of course, if they all differ in 20 characters, if you go out and do some bird watching or some botanizing, you check out the plants, and you find 2 plants that are different in 20 different characters, well, you'll say, well, they belong to different genera. Can we see where the potential is? Surely an enormous amount of DNA is needed for all this hidden information. How can this possibly be? Well, not at all. This means of creating enormous amounts of biological variation. Absolutely brilliant. We would expect nothing less from the person who designed it. It's absolutely brilliant. All that is required for the vast number of species in all of their variation and uniqueness is simply 2 alleles at each gene. 2 alleles at each, we automatically get that. We get one from mum and one from dad. So we don't need to imagine there's enormous amounts of DNA. The standard amount is fine, thank you, for an enormous amount of species. Ah, well, that's not fair. I mean, this doesn't sound right, yeah? Can major genetic potential be hidden? Well, it definitely can at the trait level, with dominant alleles and recessive alleles. I'll show you another example of that. It can at the character level. I'll tell you a little bit more about that later. And also at the cell level we are very familiar with. When we were very young, a long time ago, you might be able to remember back to when you were a zygote. Yeah. You were just one cell type. But in that cell type, in the genome, in the chromosomes of that cell, was all the information for all of the different cell types you have now. It was simply not revealed in the zygote, but the information was there. It's also true at the organism level. I'll show you a nice example of that. But let's just go back to the trait level, shall we? If you go out into the wild, you can find then the wild mustard. It's over here on the left bottom. It's the wild mustard. And if you select for traits which you're interested in, you can make broccoli, cauliflower, collard greens, Brussels sprouts, kohlrabi, carrot or cabbage. All of the information for all of those different vegetables is present, sat in the wild mustard. It's simply not expressed. Or what about all the dogs? All of our puppy dogs. I've noticed that there are a lot of puppy dogs here in Malmö, yeah, which is a lot of fun. We also have a dog. My wife, she has an old English husband, so she got an old English sheepdog. I don't know if I should be flattered or not. They are beautiful dogs. All dogs are wolves. And from that genetic information, all of these different breeds appeared. What is amazing about this is the genetic differences in all of these different dog species. There's less genetic difference between all of these than there are between wild species, individuals within the species of wild wolves. And there's a lovely experiment to demonstrate that. It was made at a university here in Germany. I forget which one it was. It was in Kiel. It was in Kiel. Yeah. So they cross a wolf with a poodle. The black ones are the poodles, and then this is the wolf, Guti wolf. And we're going to cross them. And do you remember what happens when you cross things? So the first generation, the children, they're kind of boring, right? So you lose some of this, the information. Well, you don't lose it, you don't see it. So these are the cute little hybrids. These are the kids. And you take those kids and you cross them, and now we should see the genetic information appearing. They're the grandchildren. Suddenly you see all that information. It was just hidden in the children. We didn't see it in them. So, can major phenotypic potential be hit? Definitely at the trait level. At the character level, it can also be hidden. And then we come to the realm of something called epistasis. And my genetic students, they get nightmares about epistasis when I question my epistasis. But basically what is happening is one gene can suppress, it can hide another gene. So you can't see its effects. And I'm sure that here, the whole question of epigenesis and transposition will be very, very important, and the role it plays here. But let's have a look at another doggy example. So we got here these different Labrador retrievers, and we got the black, the chocolate, and the golden. And what is fascinating, you take the golden retrievers and you just cross them with golden retrievers, even though the information for black and chocolate is present, you never see it. It's the situation, the golden, it hides it. It's called recessive epistasis. I'm not going to go into all of those details. But if you then cross a golden retriever with either a black or a brown, suddenly all the rest of the information comes out. You can see it again. Yep. I just said that. I won't repeat myself. But what I do want to point out is that epistasis can hide, so genes can hide other genes. So let's look at our situation we started with, our tarweeds. How could a tarweed give rise to an enormous tree? How is that possible? Can small plants like tarweeds give rise to big woody plants like these silversword trees? Well, we can do experiments on another plant, experiments which we can actually do in a laboratory. Yeah? This other plant, it's called Arabidopsis, the thale cress. And it's a very small plant, so you can grow them in petri dishes. So botanists love it because they can really work with this, and it has a relatively short reproductive life cycle. And if you inactivate two genes, just two genes, in this little tiny plant, suddenly you get something which looks like an azalea. So here's this little tiny plant. You'll find him out, and if you go for a walk, and here I'm sure you'll find him in Sweden, yeah, in the wild. This little tiny plant. You inactivate those two genes, and suddenly, poof, you get this azalea bush. You can actually see these flowers up here. And here you can see the wood of this plant, which is normally totally herbaceous. Normally you never see any wood there whatsoever. Suddenly you get it. You just have to inactivate two genes so they don't work anymore. In fact, this process has been seen in many places. The exuberant island radiations, when species hit islands, suddenly they start to produce lots of new species. It's very fascinating the way they do that. Yeah. Micronesia is a classic example of that, Madeira and Canary Islands and so on. Yeah. And a lot of that happens then, has been seen there. And there's been unexpected appearance of woody phenotypes. Yeah. That suggests that the information for woodiness, it's already in the genome, and it's just waiting to be switched on or not. So here's the prickly sow thistle that you find in the continent. It looks like a dandelion, and its leaves are a bit thicker and sort of prickly. And when that hits then the Canary Islands, boom, you get trees. Do you want to see the little dandelion? They're at the top there. All that information was sat inside the gene, just waiting to be expressed. And then there's Echium vulgare, Viper's buglows. Beautiful plant, beautiful wild plant, my favourite wild plant. I have to have favourite wild plants and favourite animals because my students always ask me, what's your favourite animal? But these are beautiful, beautiful plants. It's got a flowering panicle and from that little bunches of flowers, they're called cymes, come out, and they have pink and blue flowers. Very, very beautiful plants. When these arrived at these islands, suddenly you got all of these woody plants. This is herbaceous, it doesn't have wood, but these species do. These are quite famous. They're called the Pride of Madeira. And they're lovely, huge panicle of flowers. Or these are extraordinary. A person is about half as tall as this. So a tower of flowers. That information was sat in the genome just waiting to be expressed under these conditions. Woody members of these two genera, they're given they arose from herbaceous or non-woody flowers on the continent. Is this something special or unusual? No. It's been found to happen more than 1,000 times on 375 different islands. It's a very common process. Insular woodiness appears as a result of activating genes that are already present in the genome. So we know that major phenotypic potential can be hidden in the genome. We know at the trait level, we know at the character level. We've already mentioned the fact that we understand that at the cell level. It also happens at the organism level. There's the creepy one with our genders. I'm kind of sorry for you guys. The Lord decided I was going to be male. But actually the information for a female version of me, and beautiful professor will be talking to you. But I'm sorry, you get me instead. So. It happens in metamorphosis, where we have a little caterpillar, and that turns into a lovely butterfly. And the genetic information for the butterfly is sat in the caterpillar. It's just waiting to be expressed, but the information's there. There's another very fascinating example. This is the axolotl, the Mexican water monster. It's a very popular pet. It looks very cute. It has these gills sort of outside of its body. It's quite large. It would sit comfortably on your hand. It's not tiny in any sense. It's a Mexican. It's a neotenic salamander. Neoteny. What does that mean? That means it reaches sexual maturity, but in its juvenile state. And so because of that, its whole reproductive cycle can occur in its juvenile state. Now people worked that out, and they worked out what was going on, and they realized it was caused by a lack of thyroid stimulating hormone. So someone or other thought, well, let's see what happens if we give thyroid stimulating hormone to these animals. Boom, there's the adult. Here's a creature which technically doesn't exist. Very, very fascinating. What is fascinating is because for hundreds of generations, this potential has been hidden in this creature, and it's never been expressed until finally you got the trigger. The information was always there. It was just waiting to be expressed. Well, with all these hybrids, what can we say? What can we say about them? These matrices, what they do is they tell us that the organisms belong to separate groups. We recognize these as the Genesis kinds, sometimes referred to as baramin. And in fact, we don't have a single tree of life, but we have a forest of life. I think a popular term is bushes rather than trees. So the silver sword matrix, that was one example. They're all closely related. That is one kind of organism. Here's another kind of organism. This is the abalone. A tasty dish for those people who like that kind of stuff. If you see this animal, it's a marine snail basically. It has a very fascinating shell with a row of holes in an arc of what are called traumata. And those holes are used for various purposes. But this particular family of mollusks, they all have that arc of traumata. It's quite unique to them. There's no in-betweeners. There's nothing which has sort of one or two or something. You either get that organism or you get another kind of mollusk altogether. And this is a hybridization matrix between the different species of marine snail, of the abalones. These in red, in black, have unfortunately gone extinct. There are 55 left, and you can see through these lines that many of them can hybridize with one another. This is showing that this group is a unique group, a unique family. And there's this meme, sadly it's an ignorance meme, which says hybrids are sterile. No, they're not sterile. There are many, many examples where they're not. They're only sterile in about 50% of the cases. Yeah, in this particular case is an example. You get the red abalone, you cross it with the white abalone, and you get a pink abalone, you cross it with a flat abalone, and then you take these two hybrids and you cross them, and this is what you get. This is the 4 species. It's a lab hybrid. But very, very simple to make. Another a famous example, which is why I sometimes find it strange that Maya always insisted that species have to be reproductively isolated. He was sent by the Rothschilds, an incredibly wealthy family in Britain at the time. But they got this biologist to go to Papua New Guinea and to look for rare species of birds of paradise. Now, the reason that he was sent to look for rare species of birds of paradise is completely commercial, because these rare species, their feathers were rather unique. And the ladies used to wear these feathers in their hats. It was kind of the Vogue kind of fashion thing to do at that time. So the millinery, they had these fancy feathers, and of course the rarer the feather, obviously the richer you were, so you kind of paraded your wealth, you know, on your head or whatever with these fancy feathers. Well, these rare feathers, they were they came from some species which appear to be very rare. And so the Rothschilds wanted to be able to find out where to get hold of these feathers, so they sent Meyer, who's a very famous biologist, to Papua, to New Guinea, and he looked round for them, and he looked round for them, and he couldn't find any. And finally it dawned on him what was happening. These so-called rare species weren't rare species, they were hybrids. And so he drew up this hybridization matrix. Meyer was very familiar with hybrids. Well, since that time, these have been studied far more intensively. We know about 24 different hybrids. I don't expect you to read this, it's just to show you that there is a list of these hybrids. If you want to, you can come back later with a microscope, and read that list. These are the different species. They're placed into various species groups. The manucoids are up here. They look like crow-like birds, and the birds of paradise might well belong to the family of the crows, interestingly enough, even though they're such very beautiful birds. But sometimes when you look at crows and ravens, you think, isn't it a shame that God didn't make sort of prettier crows? Well, if you hear the call of the birds of paradise, quite often you can recognize the crow-like call to them. Whatever, these lines show the hybridization between the various birds. And I would like to just briefly, it's going to require you to put your thinking caps on, tell you a little bit about these hybrids. Now, if species are within a genus, they're quite closely related. So hybrids within a genus, I want to call them proximal hybrids because they're very close. But hybrids between these groups, outside of completely different genera or whatever, they're further apart. So hybrids that form between them, I want to call them distal hybrids. And I want to make a distinction between proximal hybrids, which are quite close, and distal hybrids, which are quite far apart. Let's have a look at an example of a proximal hybrid. So this is the Ragiana bird of paradise and the blue bird of paradise. These two species, when they cross, they give rise to this hybrid. The Ragiana bird of paradise, that's the one which is on the flag of Papua New Guinea. And the blue bird of paradise, well, they're gorgeous. If you ever want to spend a fun half an hour, a little bit of time, and you go to YouTube and you look at the birds of paradise, they're such fun birds to study. They have these fabulous courtship displays, really, really fun. And this guy, he actually hangs upside down and he spreads out his wings and he makes a funny humming sound to try and win the lady's heart. Yeah. But when these two cross, out pops this. And it sort of looks like an in-betweenie, would you agree? This is a proximal hybrid. This next slide has a lot of blah blah in it. I'll explain to you what it's basically about. But we want to look at, did these arise via mutations, which is the sort of stuff that you'd be told in a standard textbook, or did they arise via Mendelian speciation? Now, what would we expect? Well, if they arose via mutation, there would be lots and lots of mutation to make one fancy bird, and there'd be lots and lots of mutations to make another fancy bird. So if you get the hybrid, there would be absolutely gallons of mutation. We should get something really extraordinary. Yeah. But if they were caused by Mendel's method, everything would go back to the, well, technically the all-dominant or pan-dominant state, and that would be it. So what do we see? Well, this is a cross between the magnificent rifle bird. It stretches out its wings in a beautiful way and it makes a sound like a rifle, which has all sorts of interesting consequences. I won't go into all of those details, but that's the magnificent rifle bird. Here's the 12-wired bird of paradise. See, it's quite different. It's these big yellow feathers. We learnt all about feathers yesterday from Andy, thank you very much. And it has then these funny little shafts stuck out the end. And nobody knew quite what they were for. Well, these birds of paradise, they're always trying to win the lady's heart, as it were. And so what happens is the male calls the lady, and the lady arrives on the same branch, and then the male tickles the lady's beak with these. And the lady finds that really very nice. So she completely agrees with this. Very different birds. You cross them, and you get this thing. Now the hybrid appears to be, it's lost a lot of the spectacular differences here. There's none of the greens here, none of the yellows. It's lost all of these shafts. But we know the information is in the hybrid. If we selected it enough times, if we back-crossed it enough times, we'd go back to this. So the information's there, it's just hidden. So let's look at another pair. This is the Western Parotia and the long-tailed Paradigala. These are fabulous, the Western Parotias. They clear a little dance floor for themselves. Get rid of all the leaves to make this beautiful little dance floor, and then they spin round and they flash these feathers around. Absolutely extra, looks like a little pirouetting ballerina trying to win the heart of the lady. These are less spectacular in terms of dance or anything like that, but they do have these facial masks. And if you look at the hybrid, it's lost the facial mask, it's lost these fancy feathers. You can see it's going back to the the all-dominant state, which was nowhere near as much fun, and all of this information was then hidden. Well, that's basically a picture with just about all of the different hybrids shown. And the distal hybrids are highlighted here with blue, and you can see they're all going back to the same thing. There's very, very strong evidence for Mendelian speciation. We are not seeing mutations caused all of this. Well, time progresses. I would, however, like to introduce you to some cabbage white butterflies on Papua New Guinea. I'm sure you've all seen the cabbage white butterfly. If you see them flying around, you look carefully at the wings and the top. If there's one dot, it's a male, and if there's two dots, it's a female. So these are lovely little insects. Well, at the bottom, they also are basically just white and not particularly interesting in that sense, although attractive as they may be as butterflies. When these butterflies arrived in New Guinea, something happened, something was triggered, something very special was triggered. And the bottom of the wings, the ventral wings, suddenly started to display bright colours. Most of these experiments are done on this side. Sadly, New Guinea is split in half between two different countries. But the western side here, there's mountains and stuff like this. And I'm specifically looking at the butterflies on this half of the island, simply because it's not been as well studied on the other half yet. You find there are 118 different species, and these species are split up into species groups. And I just wish to show you the bottom side, the ventral side of these butterflies. The dorsal side, or the top side, is boring. It looks like, just like a cabbage white. It's got some white on with a little bit of black. But the bottom side, all of these sudden colours suddenly appear, these patterns. They're very strict, very unique patterns, and you can clearly see these different species. This is from the Dahlia species group. There are 11 species, but only 6 are shown here. And what you see here is that these species, they're always completely healthy. You find that these patterns, these traits, they quite often recur, which suggests the information is already there and is just being pulled out appropriately. You also find that they're consistently fit. You remember Darwin's argument is you have to select for the fittest. So you might expect some kind of progression in fitness here. You don't see any kind of progression in fitness. So they're always vibrant and fit. And here's another 6. Same thing exactly. And here's another 6. Same thing. I've got 118 species I could show you. I won't show you them all today. I think you sort of understand what's going on here. The observations drawn from the fact they're always healthy, that there's recurrence of these different traits, and there's consistency in their fitness, very strong proof of the fact that the information is already there. If these things had to mutate into existence, you'd expect to see a lot of pretty trashed out animals, because mutations aren't good for you. And in fact, the same sort of thing is seen whether you look at these silver sword plants, or the honey creepers, or the cichlid fish, or the dewlap lizards. So we know we can get lots of new species. How quickly does this occur? You can run the math. And if you follow Mendel's experiments, you see it only takes about 20 generations. Here's the adaptive radiation of the geckonic lizards. All these different lizards are all related to each other. It's to explain. What about dinosaurs? All these dinosaurs. Do they really require 150 million years to produce these different species of sauropod? No. Tens of generations is enough. It's caused by something referred to as loss of heterozygosity. So we have 2 different traits. Only one is expressed, but if we have both there, then everything isn't the same, because it's both. It's said to be heterozygous. But interestingly enough, this heterozygosity is lost at each generation. 50% is lost at each generation. And when it's lost, then everything becomes homozygous. It becomes fixed. It's rather like when you're bowling, everything's fine as long as the ball doesn't get into the trough on either side. Then it gets stuck and it's fixed. If a population has just the one trait, or just the other, it becomes fixed. A very famous experiment was done by a guy called Bury. He started with heterozygous flies, populations of flies, and within 20 generations they'd become either all homozygous dominant or all homozygous recessive. This follows what's called it's genetic drift. It's a random process, because each time you lose 50%, but you lose it randomly. And this shows you either, just as an example of that, it's a Monte Carlo simulation of this particular process. I don't want to go into too many details there, but I'd like to show you this picture, because what was unusual about Mendel's peas is they could self-reproduce. That means that because flowers can do that, they have both female and male parts in there. They can self-pollinate, self-reproduce. But most species, there's a male and a female. And then the fancy name for that is panmictic, or sexual population. How quickly does it occur? Interesting enough, it turns out that it's based on the size of the population. So if you just have a population with 10 individuals that you might have on an island, then you lose that heterozygosity very quickly. You gain new species very quickly. But if it's much larger than the population that you might have on a large continent or whatever, yeah, it takes the variation stays. It takes much longer to lose. And so here's an example of trogons, beautiful birds. And you can see basically they're swapping features. This is what recombination is all about. In a sister genus, then you have the quetzals, and then you have some longer feathers here and a few other fancy longer feathers. But it's easy to see how this information arises via Mendelian speciation. The same thing for the geese and swan family. Just very briefly, if you get these differences, can you get an infinite amount of difference? If you read Mendel's paper, you find he tackled that question. And the answer was no. You can only get a certain amount. Whatever information was put there in the first place, that limits just how many different species you can get. And of course the process itself is driven by loss of features. So technically you can't go beyond the group, the family, the baramin, its borders. You are fixed in and sat there. So just very brief to finish with. Now we can look at the genomes and we can sequence the genomes of these organisms. We can look at every single base pair and we can compare them. What have we found? Well, very recently that was done for the baboons, and these are all the different baboons. And you find very clearly when you look at the genomes of these animals that they arose via hybridizations. What about our fish? Same thing has been done. Many species have now been, the genomes have been sequenced. How did they arise? Hybridization between two divergent lineages facilitated this process by providing genetic variation. And as a third example, how these genesis families, these genesis kinds arise. Let's go back to the cat family, shall we? We've become familiar now with the liger and the tigon. You can get ligers and ligers and tigons and ligons, and fabulous. You can get other things. You can get leopons or jaglions, and all the information's there. It can be shared, and when it is, and they've looked at the genomes, and they found not only that it could occur, but that it has occurred in the past. And they can even now see the sections of the DNA, how they were swapped out. That was a paper that came out 2019. So what are the conclusions of all of this? Conclusions. Species and families normally arise not via mutations, but via Mendelian speciation. The genetic information required to form even the vast number of species that arise in these exceptional adaptive radiations is already present in the genome. Although mutation occurs, it's simply not necessary. In fact, it's detrimental. In Genesis 1, verses 11 and 21, 25, that tells us that God created the plants and animals after their kinds. The plants and animals God created had genomes already endowed with all of the phenotypic information they would ever require to produce a plethora of species. Meiosis and reproductive isolation, they enable these genesis kinds to rapidly give rise to numerous, vibrant, healthy species. Our God has so much to teach us about His glory and prepared His creatures to enrich us with knowledge about Him and to honour Him. Thank you for your attention.
Thanks a lot, Nigel, for an excellent talk. And
You're welcome.
showcasing how efficient Mendelian speciation is within the kinds, the bushes, the forests that God created the first generation, and then it explodes into speciation. So we do have time for some questions.
Yes, I have a question.
No, please.
If you take two poodles, do you think you could create a wolf again?
Oh, sorry. A very interesting question. It all depends how much information was lost from the original wolf as to whether you could get back to the wolf. Nevertheless, there have been attempts to do that kind of thing. What they have tried to do is with the original wild ox, the aurochs, yeah, and they literally try to select back to the original ox. Now you mention it, they also did that with the quagga. It looks a little bit like a stripeless zebra, and they've done the same thing there. They've managed to select back to it. There was another example that came to mind now, but technically yes, you could. So thank you for the question. Yeah.
Not from poodles alone.
Not from poodles alone, no.
Yeah, we have been told now and then that a lot of species have disappeared from the world. And is it a fact and is it a problem? Is it a normal variation in species? Or?
It's a real problem, and there are attempts now to redress that. Do you remember I showed you some pictures of these beautiful little birds in Hawaii, these honey creepers? Yeah, many of them have gone extinct. Avian malaria arrived at the islands and it wiped out quite a number of those species. Some really beautiful birds, unfortunately, have gone extinct, and we only have pictures of them now. But the hope is that by crossing the remaining species, that perhaps you can win back at least some of that genetic material.
Is it a problem that a lot of species have disappeared?
Mm-hmm.
And why have them? Why have they disappeared?
Well, for all sorts of reasons. Yeah, I think very often due to man not really thinking about what he's doing, and then perhaps eradicating their normal living environments or whatever. But there's a lot of potential there. With recombination to try and bring back some of these species back. I know they're trying to win back the woolly mammoth, but there they hope to get the actual genome information from frozen specimens, and then they'll put those into living elephants and hopefully. So it's not quite the same process there, but there are attempts to try to win back some of those species. Thank you.
Can you comment on things like the dodo and the kiwi, two different examples? Dodo gone extinct, but would it be possible to get something similar to that back?
Well,
and kiwi, a different question really. Is it really a mutation, or was it made flightless?
Oh, fair enough.
Two different questions.
Two very different questions. The first question, I think, ties in with the previous questions, and I'm sure that there would be attempts to try to do that using various auk species or whatever, to try to recombine them and to see if it would be possible to do that. So I don't think it's out of the question, yeah. But let's see what people come up with, the scientists come up with. As far as the kiwi, it all depends on your attitude. It seems to me as though when our Lord was talking to Job, that he was quite proud of the ostrich.
Yeah.
And even though it's flightless. And so I don't see why the kiwi should have to have been a mutation event. Yeah. But, yeah.
So, I mean, there are examples of flightlessness coming through mutation, like flightless cormorants on the Galapagos. Pretty obviously.
Yeah.
They've got tatty feathers, you know, and they just they don't grow properly.
Yeah. No, if that to me would be certainly an example of then mutation events occurring and perhaps preventing that. But there are other examples. There's a very famous parrot. I think it's in New Zealand.
Yes, there is.
Yeah, that's flightless.
The name will come back to me.
Yeah.
But you're right. There is a flightless parrot.
And once again, it might well be very advantageous. One can easily see how genetic processes could actually promote that in a positive sense, yeah, not necessarily just in a negative sense. My sense is that if it's caused by mutations, you're going to end up with scraggly birds. Yeah. But if it's actually designed to be like that.
This is why I asked the question about the kiwi. Kiwi doesn't seem to be looking as though it's got scraggly feathers.
Yeah.
It looks as though it was never meant to
Fly.
It just goes around in the undergrowth.
Mm-hmm.
But it is a bird.
Mm-hmm.
Yeah.
Mm-hmm.
It's a national bird of New Zealand.
Yeah.
Yes.
Thank you for this wonderful speech. I just found out, I'm so glad, as a Sunday school pupil, I thought that the Noah's Ark, maybe it was great, but still it's not fit everything. But if you say that they have already hidden lots of information, so it's quite explainable.
Yes, certainly. It's Mendelian speciation provides a very logical genetic mechanism and explanation for how that could happen. How many species could arise very quickly from, even if it's just a pair of cats or a pair of bears or a pair of dogs, how it could easily give rise to many species. Technically, it's not that difficult if you use Mendelian speciation, because we only have about 40, 50 species. We really don't need many genes to make an enormous number of different species. So yes, that could come out very simply from that process, yeah. Thank you for... Yeah. Oh yes.
I would like to add to that something, because I think it is very important for people who lead Sunday school classes that they read into this type of things, because then they can explain it. And it's a very simple example is the dogs.
Mm-hmm.
We have 400 species of dogs, which come from wolves. So there was only probably one dog kind on the ark. That also brought forth the foxes, the jackals, but also the dogs. So this ark can fit easily all these animals.
Yes, definitely.
Yeah.
So we have time for one last question. Yes?
I'm just thinking about the difference in humankind, the variation that we see between skin color, height, eye color, and all the variation.
Yeah, and of course, it's not for me to say how the Lord did things. But I can certainly imagine very easily that all of these differences we see, even these racial differences that we see, it was all planned. I think God doesn't like to be boring. If you look around at the different faces of the people, you can see very many differences. The Lord delights in that, and it's one of his presents to us. So it didn't make things which were uninteresting, but in humankind he gave us all sorts of different things, like different skin colours, different hair colour, different eye colours, and so to appreciate, to enjoy.
I have one question. During the conference days that we have had here, we have also been discussing epigenetics and also these kind of jumping genes and that kind of things, transposable elements. So in the material that you have presented, does that cover any of the epigenetics?
Yeah, well, early on in the talk I did mention this. Yeah, so what we need is a genetic mechanism to hide and keep this information hidden. And for that, epigenetics and transposons are fabulous. There's a lovely little example from transposons, if I may mention it. It's to do with wine. So we have red wines and white wines. And the white wines, they're won from green grapes. And the red wines typically won from red grapes. The grapes started off black, and they were very dark red. And that's caused by pigments called anthocyanins. And there's a master controller switch in the genome which switches on pigment production, and then there are quite a few proteins involved in making anthocyanins. But this switch a transposon jumped in and in fact blocked its activity. So instead of getting black grapes, you got green grapes. And then a little bit later, the transposon jumped out again. It left a little residue in, and you got red grapes. And so we see here a classic example of how transposons can bring phenotypic differences to the table. So very important as mechanisms to describe how then we can switch on or switch off genes and traits and phenotypes within a family. Thank you for asking.
Yeah, and thanks again, Nigel, for an excellent talk.
Yeah, you're very welcome. Thank you. Yeah.
And now we all look forward to the service starting 11 a.m. and Göran Schmidt, a member of Genesis, he will give the sermon also on the creation topic, I think. So welcome then, and God bless you.
Thank you.
Thanks a lot. Yeah, it's interesting to sort of see how all these mechanisms